So today I'm talking about the Gladiator project and the role that our team at the TSSG and specifically myself, what we are doing in the project. So the basic aim of the Gladiator project, it's a FET open project, and it's a very enthusiastic project where the focus is the research focus of the research is the brain tumor, specifically glioblastoma, and the whole goal is to come up with an externally controllable device for the purpose of diagnostics and also therapeutic of this type of the tumor. Now, glioblastoma is one of the very rare and very aggressive form of brain tumors, and it has a very low survival rate and a very high recurrence rate. So this type of tumor is very resistive to different type of therapeutics like chemotherapy and radiotherapy and all these things. So this project is very ambitious in a sense that it's using expertise of different people from theoretical people like us who are working in molecular communications to the experimental partners who are actually doing experiments on mice and all these things to verify the results of the theoretical part and all that. So I'll be talking about the role that we are playing in this project, and we are particularly focusing on the theoretical model of it and using molecular communications, which is sort of our strength as well. So basically, what happens in a molecular communication system is that there is this transmitter bio-nano-machine and there is a receiver bio-nano-machine, and they communicate with each other using molecules. And so the whole concept is the communication is happening within the brain cells, which is responsible for the proliferation of this glioblastoma tumor. And so this communication is molecular communication in our view. And what we are trying to do is design an external device which also uses molecular communications, but it uses the molecules that are programmed or designed in such a way that they go and interact with the natural system and reduce or stop the growth of the tumor, or in later stage it could be used as a therapeutic mechanism to reduce the tumor or destroy the tumor site. So this is a basic introduction of this project. So I'll talk about the objectives and deliverables. So the objective for our side is to develop an end-to-end communication model for the communication that's happening between the brain cells, and later for the communication between the external device and the brain cells, which are the natural brain cells. And so most of our work is the design and analysis within a simulation environment, and which are based on the mathematical models for both the intra and intercellular signaling. And so the whole aim is for us is to provide the simulation work as an accurate testing platform to support the experimental work that other teams are doing. And so we define this in three stages. That is, first is the generation of the tumor or in a single cell, and then it propagates or transfers information to other cells, and finally it proliferates to a number of different cells over the period of time, which is a very quick period of time. So the deliverables for us, which we have already sort of done, is to present a theoretical model for the generation and the proliferation of the tumor. And the next deliverable for which is in the next few months is to develop the simulation framework for the therapeutics and experimental verification for the experiments that people have been, the other partners have been doing. So this is again what I've talked about earlier, that a molecular communication system is something like this, that there's a transmitter, then there's a channel, and then there's a receiver. So this is a figure from the partners where they are working on the development of the therapeutic mechanisms for the brain tumor, and you can see the external devices there, which is responsible for the feedback and also to control, and so there's a reporting and feedback mechanism as well in their design. So moving on to what we are doing. So specifically myself. So what we are doing is to focus on the cell-to-cell or intercellular communication within the glioblastoma cells, and what we are trying to figure out is how the tumor grows over time as a result of the communication between the cells. So we have taken a classical agent-based modeling system, and what we have done is we have defined these cells as a voxel model where each voxel is sort of a cubic shape. And so what happens is there is a single cell or single input cell, which is, so we assume that that cell has already been corrupted or is a tumor cell as a result of some internal mutations of the cell, internal mutations inside the brain. So once this cell is corrupted, it communicates with all the cells that are connected to it, and it transfers the message which through the molecules, which is effectively the proliferation of the tumor that grow unnaturally. So the tumor, to define the tumor is just the unnatural growth of cells over the time, or abnormal growth of cells over the time. So the input is from the tumor cell, and the output is an action of these cells, such as growth or the abnormal growth, which can create, which can increase the size of the tumor over the time. So what we are trying to do is develop a mathematical model for this intercellular communication, and to link the information flow, or the mutual information between the cells, with the growth rate of the glioblastoma. And the next part is obviously to take the experimental data from the partners and see whether our theoretical model fits with the experimental data. So again, so this is what I've talked about in the last slide as well. The long-term goal is to use the concepts of molecular communication and to link the information flow to the action, which is the growth of the tumor in this case. So what we have tried to do is to consider the configuration of different cells. So as I showed you previously, here we have a two-dimensional sort of model for the cells, but actually cells are in 3 dimensions. So what we have done is we have tried it with different configurations of cell. This is a very simple cell in a series. This is a one-dimensional sort of setting where there is a single tumor cell, which is then communicating with the first receiver cell and then communicating to the second receiver cell and up to end receiver cells. So these molecules are actually taking the message from the tumor cell to the next cell and on. And the message obviously is the abnormal growth. So what we have tried to do is to consider different configurations of cell. Obviously, we have started with a very simple setting, which is a series setting of cell, and then we have followed it up with, okay, now what if the cells are in parallel and how this works? And the final of these is a hybrid configuration, which is actually a more realistic configuration of cells, that a tumor cell goes through and releases the molecules that goes to number of cells in either direction of the tumor cell, and then they go on and transfer this message up to n number of cells in a network of cells. So this is eventually becoming a network of cells, which we call it as a molecular communication network, which is something that we have expertise in and which we can study in context of this glioblastoma tumor. So this is an example of a 3D sort of a setting. As you can see here, we have so each of these rectangles or each of these voxels represents a single cell, and we have considered an example here where we have X and Y and Z, all of them are 4. So it's a 4 cross 4 cross 4 number of cells for us in this 3-dimensional setting. And as you can see here, there's a tumor cell, and it is impacting the molecule. It is releasing the molecules which are transferred to a number of cells which within the close vicinity of this of this tumor cell. Actually, what we are considering is the tumor cell is impacting almost all of the neighbor cells. So the overall goal was to apply the information theory and communication theory concepts to study the different communication properties of how the information is transferred between these cells and what is the impact on the growth of the tumor. So what we have done is we presented a communication theory model of intercellular communication between cells. And again, and in addition, what we have done is we have developed a growth of a tumor growth model as well, which is again a mathematical model, but that the growth of the tumor is dependent on the information between the cells. So 3 parameters we use specifically. One is the mutual information between the input and output number of molecules. And the second one is the information propagation speed, that how fast this information is transferred. And lastly, we have seen what is the growth rate of glioblastoma as a result of this information transfer. So I'll just briefly show you some of the preliminary results that we have for this. So this is mutual information for a number of cells. So as you can see, we consider 100 cells for this case, and we have considered 3 different settings as I mentioned before. One is the series configuration of cells, and the other is parallel, and the final one is the mixed one. So for now, our results are very interesting. It shows that the series setting and the parallel setting actually behaves much better in terms of mutual information as compared to the mixed cell. Now, intuitively, we can understand this because in a series setting, the loss of molecules is very low as compared to the mixed setting, because they're more likely to reach the receiver without any loss of the molecules. And so the other results I have are mutual information related to the number of molecules released by the external signal. So this is also very intuitive, that as the number of molecules from the input cell increase, the mutual information tends to increase. But I have to say this eventually plateaus. I've not included the result here, but eventually the mutual information becomes consistent over time. And finally, so this is the tumor growth rate. So also the tumor so this is against the number of cells. So when the number of molecules increase, the tumor growth rate the tumor grows up to a certain number of cells. So again, as the input tends to increase, the tumor growth rate tends to increase with it. But this also plateaus at a certain point, because there is a fundamental limit to the way molecular communication works. So unless there is a consistent source of the input, there is a limit to that. So I think that's pretty much it from me. I think if you have any questions, I'm happy to answer. Thank you.
Thanks very much, Amden. Does anyone have any questions?
Yes, may I have a question?
Yeah, sure.
I'm curious. Thank you for your study, very interesting. I'm curious about, so you want to study about the growth rate of glioblastoma cells, because you, sorry, first I asked what is the main purpose of that? And the second one is, did you check also the growth rate of the cell, of the neuron cells?
So these are so the purpose of studying this in the first place is because glioblastoma is one of the most aggressive form of tumors, and we need to find a solution for this type of tumor. So this is a step towards that okay, we are studying how the glioblastoma proliferates. So the next step of our research is to develop a therapeutic mechanism for this type of tumor. And again, we will, as I mentioned, the external device concept. So what we will be doing is we will be inserting external molecules within the cell, which within the brain cells, which are going to suppress the information that, or the information from the tumor cell, or overcome it, so that the proliferation or the fast growth of the tumor is reduced, or maybe eliminated, something like that. So this is the next step. But our main goal is to reduce the growth of the tumor, because right now what we have observed in both from experimental studies and the theoretical model is that tumor grows very fast over time. and we need to reduce that in order for the patients to have a good survival rate or a good life quality.
Right. Okay. And then did you relate that to the growth rate of the neuron cell also?
So these are actually so when we consider cells, these are in a way neural cells because they are in the brain, so they are glioblastoma cells in the brain. And we are only considering the growth of these cells specifically, not other cells. But this is what I'm saying is this is type of a neural cell.
Because I assume you want to have like mathematical models, so you need kind of standardized number for that.
Yeah, we can share some mathematical model with you if you want.
Yeah, so I'm just curious, like how you can control like if one cell grows very fast.
Now we are just studying it how it grows.
Okay.
We're not
so kind of